Mitosis was done in the last video. This video starts with meiosis. Meiosis is necessary for sexual reproduction, helps us to maintain the chromosome number in sexually reproducing animals. In adult human, that is 2n, 2 number, 2n DNA content, and 46 and has 46 chromosomes. That and this is present in the pairs. That is one pair. That is one pair, that is two number of every single chromosome. And this in this structure meiosis occurs so that the chromosome number is reduced to half, so that both the parents half combine to form one of the offspring. Two gametes of one of male and one of female will fuse together again to maintain the same number. Okay, this is this will become half, this would also become half, half and half on fusion will form complete. Meiosis is a reductional meiosis since it is also leading to reduction of chromosome number, since chromosome number become half of what they were earlier, 23 from 46, and this is necessary as haploid cells formed will restore the same chromosome number as parent cell that is diploid after fertilization. It is also called as paradox of meiosis, that is reduction. Paradox here means reduction in chromosome number is leading to maintaining the chromosome number in the species during sexual reproduction. This is the at G1 the chromosomes number is 2C, and the DNA content is 2n is equal to 8. um, and then at S or S2 G2 we know that they already done 4C and then 2 at same, but in mitosis, okay, this would again return back to it, but here 4 will be 4 following would be formed, and then these 4 would again be divided into two pairs, okay, in the next offspring, so as to maintain again the same number, that is 2C. 4 again they will be divided into two, okay, these would be in pairs, so one would be two in numbers, okay, every type will have two of one more of its kind, so that is two per type, so that would reduce its number to 2C. And similarly here also in the n, in the 2n is equal to 8, whole genes are in two copies, that is two copies, okay, all genes are present in two copies, but here then there are four chromosomes having all genes in one copies. Here on division n plus n is equal to 4 and then plus n is equal to 4. Here copy one one gene copy will have four chromosomes, but here two gene copies will have eight. All matlab would be in two copies, here all would be in one copy since meiosis occurs. Now homologous chromosomes is pair of chromosomes having same gene locus. Done. These all are homologous chromosomes. All these four are homologous chromosomes. Here if you see, these are representation of 2n is equal to 8, since the total number of chromosomes here are 8, and then one type, every type has two of its ends of 2n, and then if we could talk about chromosome number, then 4C, one chromatid number, 4C, 1, 2, 3, 4, and these are 16, one, that is 4 into 8, 2 into 8, 8 pieces, and each has two chromatids, so 16, 4C equals to 16, and these are n plus n barabar 4 and plus n barabar 4, that is 2n barabar 8. Okay, this is all. And now in a diploid cell, and this is meiosis 1 agree, okay, later it is discussed. In a diploid cell, diploid, uh, every gene will have two alleles, so that every homologous chromosome has one each allele each. Now each allele for a gene is inherited from each parent, simple. And meiosis 1 when occurs, it will form two daughter cell, n plus n plus barabar 4 and 4C barabar to 8 pg. And then on meiosis 2, that is again having meiosis on the daughter cell found on meiosis 1, there would be n barabar 4 and 4c barabar to 4 picogram that would be formed. This condition will have all the genes in one copy. This one in only one single copy, all the genes, that is um, all genes would be present. Heterotypic division: genetically, four daughter cells will be different from each other. This is due to crossing over. Crossing over is exchange of DNA segments between non-sister chromatids of a homologous pair. See here, I'm crossing. This is T capital T capital T. This is R capital R capital R. This is small t. This is small r. Okay, crisscross or the change in the crossing over exchange of DNA segments. These are capital and small r. These two segments is happening between the non-sister chromatids. These are non-sister chromatids, not the chromatids of the same chromosome. And it will form a heterologous pair. This would be formed. This would be first meiosis after. Okay, so this type would be formed. T and then R capital R small capital R small r, then t capital R small r, and then again on second mitosis what would be formed? T R on this chromatid, then T capital small r capital small t capital R and small t small r. All of this is this. Crossing over occurs by breaking and reunion, which involves several enzymes such as endonuclease, ligase, U proteins to stabilize the complex. These are R proteins for recombination. There are R proteins for recombination, U proteins for stabilization, and enzymes such as endonuclease and ligase. Only this. Now steps of meiosis. Meiosis 1, then interkinesis, and then meiosis 2. Between meiosis 1 and 2 there is a short phase called as interkinesis. It may or may not be present during interkinesis. Number of DNA replication occurs without S phase. Okay, only DNA, no DNA replication occurs. So in other words, it can be said that no actually S phase is occurring. Now also interkinesis has no DNA replication. RNA is synthesized in this phase. Protein is synthesized. Organelles duplicate as well as the centrioles duplicate. Now meiosis 1 will have prophase 1, metaphase 1, anaphase 1, telophase 1, and meiosis 2 will have prophase 2, metaphase 2, and anaphase 2 and telophase 2. Just naming, and after the name there would be 1 or 2. Now meiosis 1, that is the first one. Of course, animal cell has 2n barabar 8 at the first, after at S stage or G2 stage, 2n barabar 8, that is 4c barabar to 16 picogram. Meiosis 1 is the phase for separation of homologous chromosome. It is also a phase for crossing over, that is variationing. It leads to reduction in the chromosome number, that is haploidy of chromosome. So and then again prophase 1, that is the first one. Now this is the longest stage and it has further 5 phases, that is leptoline or leptolemma or zygotine or zygonema, or third one is packetine or pachynemma, and the fourth one is, yeah, here, diplotine or diplonema. Nema, lean. Now, first one is leptoline, that is DNA starts to condenses here, so that the threads of DNA are so that the threads of the DNA now become distinct. These are the threads of the DNA, and they become distinct now. Okay, these threads, and the special thing is that the DNA, condensing DNA appears like beaded threads. Such beads are called chromomeres. These beads, circular things. DNA further condensed around the nucleosome is called having actually chromomere. And now the main thing here is that the DNA appears to have its free ends toward the chromosome centrosome. That is, there are two pairs of centrosomes. Therefore, so these will have the free ends of that is the start and that is the end of these chromosomes will be near the centrosomes. Now, so this is called Banker's stage. Now, zygotene or zygoderma, this DNA condenses further so that the chromosomes are now visible as long thread. The homologous chromosome will start to align parallelly. Now, this arrangement is also called a synapse. And the chromosome appears single-stranded at this stage. Now, as the condensation is not yet complete, sister chromatids of each chromosome are not distinct. Each chromosome therefore appears single-branched, though they are distranded. Such a homologous pair of chromosomes is called bivalent, that is two-stranded stage. Through homologous pair is actually four-stranded. Okay. In a synapse pair of chromosomes at several places, proteinaceous bridges are formed which hold arms of non-sister chromatids closely. Such regions are called synapto-lemon complex. At these regions, crossing over may occur. These are the cytonymal complexes, and it will have euproteins, endonuclease, ligase, and recombinant exam. Recombinant exam here is for controlling process of crisscross. That is the heterozygous formation, heterozygous chromosome formation. Now hmm, next here we have packaging or pachynyma, and the DNA condenses further so that the homologous pair of now appears to be tetrad, that is four-stranded. This is a tetrad set, that is four-stranded. Now the points where the synaptonumal complex crossing over occurs it forms a recombination nodule. This is all about packeting, nothing else. Now diplotin is an important stage. DNA condenses further here. The synaptonumal complex where the crossing didn't occur will dissolve away, and the regions where the crossing over occurred, that is recombination nodule, now appears distinct like a cross-like structure now called chiastema. Chiastema. Now the diplotin stage can be of variable duration. In case diplotin stage is spread over a long period of time, then it is called dictiotin. Now example is oocytes in human female. What it actually means, the last line is that in the human female at the during the birth only there are thousands of the oocytes, that is egg cells, and after 15 years of age they start to be used per month. Okay, and the last one that is released at the age of 55 has to be okay, stored in the body since the birth for 55 years. So that is how a long period of time is talking about dictiotin. That's all.